I drew the cell membrane for you, and we talked about phospholipids together making the cell membrane. I told you the cell membrane has proteins in it, and I told you that those proteins are important for the movement of sodium and potassium ions. But here's what I said. These are charged ions, which means the cell membrane, which is a very non-polar or non-charged structure, resists these charged things, which means the sodium and potassium cannot go through the cell membrane unless there's some kind of hole specific for those ions to go through. And I said those holes are made out of protein. They're channels that allow the charged ions to go in and out of the cell. And so by logic, you should have been able to get to the fact that since there's more sodium outside the cell, it would diffuse in. And since there's more potassium inside the cell, it would diffuse out. Logic should have told you it goes from higher to lower concentration. What you did not know, and it's my fault, is that the reason the sodium and potassium are distributed this way is also because of protein in the cell membrane. This one, as it turns out, will become for us, this one will become for us the most important protein in all of human physiology. So I should not have missed it. This is the sodium-potassium pump. So in the processes of cellular transport, every student learns in elementary school that things go from higher to lower concentrations. If you have a balloon with air in it and you let it go, the air, the denser, higher concentration inside the balloon, will come out of the balloon. Simple diffusion. Everybody learns this. Things move across membranes from higher to lower concentrations. That's the nature of things. But sometimes we can move things against their concentration gradients. If you want water to go downhill, you stand at the top of the hill and you pour the water out and down the hill it goes. That's diffusion, really, going downhill. Higher up here, lower down there. However, if you have water at the bottom of the hill and you want to get water to the top of the hill, you need energy to do that. You're going to have to put it in a bucket and haul it up the hill, or you're going to have a pump that will pump the water up the hill. It won't go that way naturally. Same thing works in human body cells. If you see that sodium is more concentrated on this side of the cell and there's a channel that lets the sodium go through, or if you see that potassium is more concentrated on the inside of the cell and you see a channel that lets it go through, well then how in the world are you going to explain how the cell maintains this? Right? Over just a little short period of time, the concentration should be equal on both sides. Were it not for the function of this protein, the sodium-potassium pump. These are pumps that are proteins that sit in cell membranes that pump sodium out and pumps potassium in. The sodium-potassium pumps, arguably the most important protein in all of human physiology. I should not have left it out during our last class meeting. Okay, more about proteins here today. The last organic macromolecule to discuss is the nucleotides, the nucleic acids, which are built out of nucleotides. And so I'll just say a brief word about DNA and RNA here, and then I want to teach you a sketchy outline of the process by which proteins are made by DNA, and then a little story to tie them together. So first, the structure of DNA. You can see that DNA is built out of what we call nucleotides. A nucleotide in human biology is simply a three-part construction. It is built, a nucleotide is built simply out of phosphate, sugar, base. These are nitrogen bases, nitrogen-containing bases. So I'm going to say that over and over again. Nitrogen-containing bases. They are phosphate, sugar, nitrogen-containing base. So whether you're talking about DNA, deoxyribonucleic acid, or RNA, ribonucleic acid, these organic macromolecules are built out of nucleotides, and they're very similar in structure. So this is a structure of a very short version of the structure of DNA that shows you a nucleotide built out of adenine, which is a base, and another nucleotide over here built out of thymine. As it turns out, there are only 4. Adenine, thymine. Can y'all tell me the other 2? Guanine. Good. Guanine. Cytosine. Yeah, so these are things that we learn very early in our lives. You should have picked this up much earlier than when you got here. So this is the base structure of the DNA molecule with the nucleotides and the bases. Now, the molecule itself is a huge molecule made up of over 3 billion base pairs. So the DNA molecule is a double helix, as you learned early in your life, in which the bases interact with each other and the molecule twists to form a long chain which has sides made up of sugars and phosphates and interiors made up of the bases. So let me show you the previous picture again, and then we'll look at this one more time. So this is just one of those interactions. This nucleotide is hydrogen bonding with this one. So this is one piece of the DNA where this nucleotide base pairs with this one. And you can see that adenine is base pairing with thymine. And as it turns out, that always happens. So if you're going to build a new cell, so one cell is going to reproduce itself, we call that mitosis. When that happens, you have to copy the DNA. So how do you know that the copy of the DNA will have the same sequence of bases that the original did? And the answer is in a process called semi-conservative replication. You learned this also very early in your lives. If you unwind this thing so that now all you have is this nucleotide on this side, you know what nucleotide is going to be added. It's going to be this one, because thymine will always base pair with this one. So the 3 nucleotides that come in the copying process are very predictable. And the cell doesn't normally make very many mistakes. In fact, there are mechanisms in place to prevent those mistakes. That would be a genetic mutation if you put the wrong nucleotide there. A point mutation, if you will. So this is the base structure of DNA. Now, this nucleotide will have another one here, and another one here, and one here, and one here, until we get to 3 billion. It's a big molecule. So if you look at the structure here, these dark purple and light areas and dark purple lines represent the backbone. And that backbone is this. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's the rungs of that's the sides of the ladder. The rungs built out of the base pairs in the middle of the ladder are the base pairs interacting with each other by hydrogen bonding. So that's what it looks like. And it's twisted. It's a double helix. And many of you have heard the story of Watson and Crick and Rosalind Franklin and the X-ray crystallography, the discovery in the 1940s and 50s. They won the Nobel Prize for their description of this molecule. The DNA molecule. Adenine, thymine, cytosine, guanine, sugar phosphate backbones, and hydrogen bonding between the base pairs. Now, this is a pretty simple molecule, really. I mean, compared to a protein, this is a simple molecule. It can be very easily described. Its structure can. And so there was some resistance in the scientific community upon its first description as the house of the control center. That is, this molecule sits in the nucleus of the cell, and it dictates what happens in the cell. Well, how in the world is something, it's big, I'll grant you, but structurally it's quite simple. How can something with such a simple chemical structure give instructions for all the things that have to happen inside a cell? Now, you know the answer to this question already. You didn't realize you did. The complexity of life is not in the DNA molecule. Where is it? We talked about this last time. Where's the complexity? Alpha helix, beta pleated sheet, tertiary, quaternary. What am I talking about? I'm talking about the structure of proteins. Now, as it turns out, the main job of the DNA molecule is to give the cell instructions for how to build the proteins. The proteins are constructed because of instructions given to the cell by the DNA. That is what we call in science the central dogma. The central dogma of science. DNA gives instructions for the formation of proteins. In fact, we might take that one step further. What do you mean when you say a gene? You say, well, the genetics run strong in my family. I look like my dad. If you look at me, picture at my wedding, I've got this picture at home. Picture my wedding when I got married. You look at the back of my head standing there, and you take the black and white photo of my dad when he married my mom, and you put them side by side. You cannot tell whether I am him or he is me. We look identical from the back. You say, well, the genetics run strong in my family. If you guys were to meet my dad, and they'll probably come over for a game or two this fall, and so come up in the stadium and say hello. It's striking when you see the two of us. And he still looks like a very young man. He's 76 years old and doing quite well with his health. And so people come up and they like to rib me about it. You know, they say, Wade, is this your older brother? Right? And he loves that. We look so much alike. You say, well, the genes run very strong in your family. What do you mean by that? What do you mean when you say genetics? Well, this is what we mean, the central dogma. So ultimately, questions like that have to be explained in terms of the complexity of the proteins that are built. So I tell this to students every year because it sort of breaks it down to the base level. The structure of my ears here, they are built out of connective tissue mostly, cartilage. And the cartilage that is in my ear has protein in its matrix that determines its particular shape, its arrangement, where the bumps and the turns and the lines are. Those are all dictated by proteins that are in the matrix of the connective tissues. So the biggest protein in these connective tissues is a structural protein in the human body called collagen.